Hello young guys, you are welcome in the zoology class organized by Jan Vikas Mahasabha and Society of Career Technology. Today we will start about physiology of digestion. Before coming to the point, I would like to emphasize you how the concept of digestion developed. Actually, all informations of the science, they are an outcome of some experiments. I remember that there is a book, Basic Physiology by Dayal. He had described that there was a old man and so every morning one of his 12 years old son used to collect empty bottles of different drinks and used to throw it in the dustbin. One morning he observed that there was some half teaspoonful remaining in the bottom of the bottle. As it is very much natural, it is obvious. That boy just tasted that and he enjoyed it. So he developed a habit of taking that small spoonful of drink which was left in the empty bottles. One day, the person responsible for cleaning of the washroom brought an acid bottle, used half of it, half was remaining. He just kept on one side in the corner that bottle. That boy was not knowing this. He just thought that it is also some drink which was left last night by somebody. He just drank it. And you know what would have happened. His all mouth cavity, esophagus, half part of the stomach was burnt. And only option was that the doctor operated his stomach and connected with the belly wall, made an opening of mouth here. And every day the food was given to that boy from this mouth. One day, one of the friends of his father arrived. He was working in a circus. He just carried that boy with him. And he used to show this to the children coming to see that circus. He used to tie a fish in a thread, used to put in the stomach. It was digested. After some time, he used to take it out and show that, well, this much part has been digested. So this has been written in the Basic Physiology by Dayal. Actually, the unicellular organisms, for example, amoeba, had a system that when a food particle came in the vicinity of the cell membrane, a small bushing was formed, and after that it was invaginated to form a food vacuole. A lysosome fused with it to form a food vacuole, and in this food vacuole the food was digested by the activity of those lysosomal enzymes. But a big animal like we need much amount of food for the need of every body cell. It is not possible that like an amoeba we should drown in a bathtub and rice, pulse, etc. should be soaked in that water and can be absorbed from our skin. In multicellular organisms, there should be a system where the food can be broken down into such smaller pieces which can be utilized by the cell. So what happened? A cavity developed in the middle of the body, the alimentary canal, and this alimentary canal food was broken into smaller pieces, such smaller pieces which can be utilized by the cell. It was taken into the blood and blood carried that digested food into every body cell. So this way an alimentary canal developed. Embryologically also, the alimentary canal is made up of an endodermal layer in the middle. The outer body wall is made up of ectoderm, and it is interesting to note that some of the ectodermal cells invaginate through the mouth opening into the cavity of alimentary canal. Similarly, some of the ectodermal cells also transferred from the anal end. So, three parts were in the alimentary canal. This part, which had an ectodermal innermost lining, was called foregut. This part, which was not having that lining, was called midgut, and that this part, which was having again ectodermal lining, it was called hindgut. The mesodermal cells arranged in the two lines, one around the alimentary canal, which was called splanchnic mesoderm or visceral peritoneum, and the other layer just below the body wall, which was called somatic mesoderm or parietal peritoneum. And now a cavity developed in between these two mesodermal cells, which were called coelom. So coelom is a cavity which is surrounded by mesodermal cells from both sides. Now let us see. Now, one very interesting part is that in your syllabus, transverse sections of different parts of alimentary canal are shown, and every time you just try to remember different layers of that. It is not the system. Actually, you have to learn that the basic pattern is the same all over in the alimentary canal. There are four layers. The outermost layer is serosa, derived from splanchnic mesoderm. This serosa layer is absent from first half part of the esophagus. After that, beyond the second half part of the esophagus, we have that complete layer. After that, there is a layer of connective tissue called subserosa. It has two layers. The outer layer is longitudinal muscles, and then inner layer circular muscles. And in between runs a part of the nerve fiber, Auerbach's plexus. After that, we have the layer of submucosa. Submucosa is the layer of connective tissue. Here also a nerve network runs through, which is called the Meschner's plexus. So these three layers are same all over in alimentary canal, but only thing is that the innermost layer of mucosa is different in different parts. In esophagus, it is simple, having only some mucus cells. In stomach, they have gastric glands. In the duodenum, they have Brunner's gland. In the small intestine, they have crypts of Lieberkühn. So, in different parts of the alimentary canal, only thing is that this innermost layer mucosa is variable. And there are different parts of alimentary canal, you know. The mouth, which enters into the buccal cavity, part in between our lips and the teeth is called vestibule. After that, there is pharynx. Here in buccal cavity, we have three types of salivary glands which secrete saliva. Esophagus has no digestive enzyme, but it secretes some mucus. Then in stomach, some gastric juice is secreted. It leads into intestine. Intestine has two parts, small intestine and large intestine. Small intestine has three parts, a U-shaped duodenum, then ileum, jejunum, and then ileum. The large intestine also has three parts, colon, rectum. Here we have a finger-like process, vermiform appendix, which is much more functional in herbivorous animals. Here some cellulose digesting bacteria are found. In human being it is not non-functional. It exists as a vestigial organ. And then there is a rectum which opens outside through anus. Regarding the biochemistry of different digestive juices, we will discuss tomorrow. Thank you very much.